science fans, and welcome to SHE-ensya. Our topic for today is the practical applications of genetic technology. For a lot of people, science is something abstract and not connected to the real world. So, for this video, I will be showing you how to connect our pagka-addict sa research into real-world applications.
Baliw na baliw kayo sa research. Aanin nyo ba yung research?
Personally, I'm most comfortable working with DNA. And for the past 8 years, we've been working with mangrove crab fishermen or alimango fishermen in order to improve their productivity. So, where do we start? First, we have to get the DNA away from the protein nucleosomes, then pull it out of the nucleus, and then finally out of the cell. The first thing we do is to break the tissue structure of the source of DNA. I'm using Indian mango, and the chopping of the mango is a physical way of breaking the tissue down into smaller parts. This process is called homogenization. Once we're ready to separate the cells within the tissue, we need to first place it in an environment where the DNA won't break down. In the lab, we get to place our samples in a buffer that controls the pH of the environment. For now, we put our samples in a saline solution. The salt helps neutralize the charge in the DNA, making it less soluble in water, which will be extremely useful later. Now, we continue the homogenization of our samples mechanically through blending. Once the cells are fully homogenized, we add dishwashing liquid, mix, and let it incubate for a few minutes. In the lab, we use more powerful detergents such as SDS for DNA extraction. But why use detergents in the first place? Detergents interfere with the structure of lipids by attracting the nonpolar ends of phospholipids, causing them to aggregate and eventually dissolve. Lucky for us, the cell membrane and the membranes of the nucleus, the mitochondria, and the chloroplast in plant cells are all made up of phospholipids. And these are the structures inside the cell that contain the DNA.
A few moments later.
So, after waiting for a few minutes, we can then add pineapple juice or contact lens solution to destroy the proteins in the nucleosomes. A nucleosome is a section of DNA that is wrapped around a core of proteins, allowing it to be compressed into a small volume inside the nucleus. The core of the proteins are called histones and must be destroyed during DNA extraction. We can then add isopropyl or ethyl alcohol slowly along the sides of our container. Since DNA is not soluble in alcohol and is also less dense than alcohol, DNA will ideally float to the surface. So once we have DNA, what do we do next? Well, in our case, we look for unique sequences present in the DNA of certain organisms to confirm their species or their identity. We do this by cloning the natural cloning process of the cell or DNA replication. In the lab, this method is called polymerase chain reaction or PCR. But instead of copying the whole DNA, we only copy the portions that we need. PCR is done by mixing the extracted DNA with a cocktail of substances that will mark the beginning and end of the portion you wish to copy, attach the counterpart nucleotides to the portion that you need copied, and to do this several times until you have billions of copies of the portion that you need. Fun fact! If you're working with bacterial cells, you can actually skip the whole DNA extraction process. And this is because the cellular structure of bacterial cells are so simple that they pop open during PCR itself. A PCR cocktail will generally include the following. A PCR buffer. Buffers contain substances to stabilize the pH to keep DNA stable and the enzymes involved functional. A free nucleotide mixture. This mixture contains free nucleotides that will be used to create the new DNA strands you are copying. Forward and reverse DNA primers. To mark the start and end portion of the DNA region you wish copied, primers also trigger the action of the most important component of the PCR cocktail, which is the Taq polymerase. By the way, primers don't just come with any PCR kit. You have to get them tailor-made. What you need to do is to send a sequence to a manufacturing company, and you can get the sequence either by designing the primer yourself or copying from a published paper. There are generally two types of primers. A universal primer that will amplify sequences present in a lot of organisms, or species-specific primers that will only be amplified if the tissue you're working on matches the species to which the primer belongs to. And, as mentioned earlier, the most important and most expensive portion of your PCR, the Taq polymerase. Taq polymerase behaves like a DNA polymerase and attaches free nucleotides to their partners in the DNA region being copied. So, the first part of actually doing PCR just involves almost like a cooking show with pipetors. You just make mix different solutions. The hard part is computing the correct volume of the needed substances to make a master mix, and making sure that you pipette accurately to make sure the PCR works. Once you're done preparing your PCR cocktail or master mix, you can then load your samples into the PCR machine or thermocycler. This machine facilitates the different phases of PCR by creating fluctuations in temperature. The first phase of PCR involves denaturation that unzips the DNA, just like how the enzyme helicase does it inside the cell. This is then followed by repeating cycles of denaturation, annealing, and elongation. Annealing allows the primers to attach to the correct complementary region, while elongation is when the Taq polymerase does its job and copies the target portion marked by the primers. The final elongation is to allow the Taq polymerase to complete copying the entire length of the target region. By the end of a 2 to 3 hour PCR run, you will have millions, even billions of copies of the DNA region that you wish to study. To confirm your success, you run a portion of your PCR solution into a process called Agarose gel electrophoresis, where electricity is used to make the negatively charged DNA fragments move across a gel matrix to separate them by size. By looking at the agarose gel under UV light, you will be able to check if you are able to amplify the correct region. This is done by looking at the size of the DNA fragments amplified. The higher up they are in the gel, the longer they are because they moved slower through the gel matrix. The lower down they are, then they are shorter because they were able to travel fast through the gel matrix. The sizes of the fragments can be approximated using a DNA ladder, a mixture containing fragments of DNA of known sizes. If your PCR product is of the correct size based on your agarose gel electrophoresis results, then you're good to go. Successful completion of your PCR would mean that you have samples ready to send off for sequencing. Traditional sequencing, also called Sanger sequencing, uses a modified PCR approach to identify the sequences of DNA base pairs in your sample. The result of sequencing is a document containing AGCTs in different sequences showing you the content of the DNA you sent out. And we use the unique sequences found in these molecules to find a match in existing databases. One of the biggest databases of DNA sequences is hosted by the National Center for Biotechnology Information, and you can access this database for free. Apart from being used for species identification, unique sequences of DNA can also be used to determine the evolutionary relationship between two different organisms, as well as detect the connectedness of different populations across a large geographic scale. Also, a modified version of PCR is even used to confirm the presence of the COVID-19 virus in tissue samples. Using DNA extraction and PCR, with the combination of image analysis and machine learning programming, we were able to create a free mobile application called Crabifier that identifies the species of juvenile mangrove crabs. Since its deployment last August 2019, we have helped fishermen ensure that they are capturing or purchasing their preferred species. We've also designed the pipeline of our processes in such that they can be applied to a variety of organisms. So someday, we hope to be able to do this from the farm to the pond, from the ocean to the forest. I hope you were able to learn something from this short video on lab practices involved in genetics. I hope you can give this video a thumbs up and also subscribe to our channel. If you have any comments, questions, or suggestions, please don't hesitate to message me, your resident Filipina scientist, in the comments section below. Thank you very much and see you around!